Welcome, come on in. Welcome to the EFL. My name is Doug Thompson and I'm one of two greenhouse curators here. And basically my job is just to do whatever's needed to help teaching and research here at UNBC. It's not just the greenhouse, I do all sorts of other things. And we'll, as we go through the tour, I'll talk about those. So we're standing in the main hallway right now, and as you can see down the hallway here, we have these big green boxes here. These are our growth chambers, 3 of which are like brand spanking new. We could actually take a look inside. These are basically like a small controlled environment. So when we open them up, nothing's actually happening in this one right now. And you can see how shiny and new they are. So we have some shelving units here. We can raise these up and down, or we can take them right out and have our plants, or whatever we might need a controlled environment for, sitting right on the pan at the bottom here. Stands for air circulation. Here we have what's known as a quantum sensor or a PAR sensor. And so this little guy here will actually measure the photosynthetically active radiation. You also have a light canopy here that you can raise or lower, so if you want more intense light, you can do that. So this guy here, close this up, is all controlled by, it's not on right now and it'll take a while for it to warm up, so to speak. But using this interface here, we can control temperature, humidity, the amount of light, the timing of light, so you can basically try to mimic some sort of environment that you want to do. So really nice, small scale, but highly controllable. In the greenhouse here, we have 4 compartments. You can see here. This is a contract with BC Timber Sales, Seedling Services. And so what we're doing is kind of like one of the last tests that Seedling Service does with the stock that they're going to be using for reforestation. And so what we do is we receive samples from all the cold storages around the province that are presently holding trees waiting to be planted out come this spring and summer. And so this is an example. A sample is like literally only 16 trees. And this could represent like 400,000 trees. I think the smallest request key that the ministry has is like 300 trees, and that's grand fir. And the largest one that they have is something like 600,000 trees, and that's actually large, which is actually surprising to me. So yeah, what we do is we literally just we'll grab a sample from the cold storage, and we'll pot up these samples in straight peat. We'll grow them out for 2 weeks, and then we unearth them. And actually, there's some students in the back today doing some work on these, doing the assessments. But you'll see that after 2 weeks, you start to get a little bit of root growth. That doesn't actually look like a ton to me, but you only know until you actually unearth them or unpeat them, so to speak. So we grow them just in straight peat, no fertilizer, nothing. We just want to give them the right amount of moisture, and we want them to just see if they're going to basically come out of the gates charging when they thaw out, essentially. So we do 660 request keys for the ministry, or tests for the ministry, and then we do a few other samples for some private contracts like Tolko, West Fraser, and some of the nurseries. To do this work, I'm mostly hands-off. We hire students to do this intent, so you can come talk to myself or the other greenhouse curator, whose name is John Rodowski. Hopefully we'll meet him in the office later. Yeah, so that's what's going on in this compartment. On this table here, we've got some strange looking plants. These are actually owned by Darwin Coxon. But what he's got going on is really interesting. These plants from Tasmania, New Zealand, Australia, so plants from sort of low southern hemisphere. He's referenced these or called these plants from kind of like the podocarpous type. And right behind you there, you can see quite a large one. This one here is Ericaceae. I can't family. So this is actually related to like monkey puzzle or Brazilian pine. And so pretty interesting plants that you don't regularly see. And then what we've got over the back here is sort of mostly orchids. And then over here, quite a strange collection of things going on here. Underneath the table, I said earlier that we just try to make stuff happen. Well, underneath here, we've got these little containers. And in the containers, what we have here is we've got a mixture of hemp, some hemp like fibers like that basically that were basically just waste fibers after the hemp seeds were collected by Hope Farms. I believe they're growing hemp out there, hemp seed. So they're composting the hemp, adding it to all sorts of other amendments, and then trying to grow edible mushrooms on them as sort of like a secondary agricultural product. And so this is an undergrad project. The student Jasneek, she's working with Lisa Wood. And so we often, it is a research facility, but we often have undergraduate independent studies in the EFL that we help with. And so this is kind of a unique one. We haven't grown a lot of mushrooms in the greenhouse. We don't really know how it's going to go. It's a hard environment to maintain, the environment that mushrooms require, especially being up here in the north. We're a very dry climate, and we don't have a lot of humidity control in our main compartments. So we put them down low, and where the humidity will naturally be, the relative humidity will naturally be higher. And we also have shade cloth here just to keep the light off. Just a collection of things here. This is one of my little pet projects. I received this seed from a fella down south. He's from Scandinavia. South being, he lives in Vancouver, but he's from Scandinavia. And this is kind of a Scandinavian curly birch. And this is prized by wood turners. And I don't think I'll be getting any wood from these anytime soon that I could use for wood turning, but I thought it'd be really a fun thing to grow. We have some native rose. This is a project that Ali Dolt is working on. Again, she works with Lisa Wood, and they are looking at the effects of glyphosate on the physiology of native plant species. And so we just pulled these out. They were overwintered in our compound, and we got them out of the snow just 2 weeks ago, and they've already flushed out quite quickly. We're here. We have a collection of ornamental stuff. This is lemongrass right here. Also, this is being grown for the David Douglas. But there's also a lot of other stuff like petunia, bacopa. We do a lot of plant starts, and we use those to make our hanging baskets that we see around the university. So that's another little side thing that John and I do off the side of our desk. We used to do more plants for convocation, but right now convocation is not what it used to be. So we might just have pictures of plants. I don't know. This is the last compartment of the EFL, and in here you can see a whole variety of plants. This is what I call kind of our show and tell collection. A lot of these plants will be used in labs for plant systematics, plant biology, introductory biology, forest biology. So what we like to maintain a collection of plants that show different adaptations to different environments, or even different plants that have different cultural significance. Over in the back there, in the succulent section, we have peyote. Yeah, so a variety of succulents here that we're panning across. This one's always an interesting one. The elephant's foot, quite a bizarre. I guess that we call it form. I'm not quite sure what that structure is really. Here's our peyote. So again, this one's an interesting one. This one here. This is what you would call like a carrion plant. We often get the students to smell this one when the flower is in bloom, because we love to see them get disgusted with the terrible smell. It smells like rotting flesh, essentially. So I think it's meant to attract flies for pollination, but it's pretty disgusting. Always fun to get the little kids to smell it. They're like, oh my god! Anyway, so orchid, cactus, hasn't flowered yet for us. So it's kind of funny, we've got, this one's here, this is an epiphyte, actually, even though it's a cactus, it comes from a completely different, environment that we consider, cactuses to be from. This one here, also very interesting, one of the very, very few cactuses that actually still has leaves. So you can see underneath there, little spines there. So, yeah, quite interesting from an adaptation point of view. We've got Brazilian pine here. Some bromeliads, ancient plants, cycads. So, and we've got it all jammed into one place, one environment. So, you sometimes have cactuses right next to, things that are like meant to be growing in the tropics. And so, it isn't the perfect solution, but we do the best. We have, the cactuses against the wall where we can, and where it gets cold at night, but warms up during the day, kind of something like you would get in a sort of a desert environment. And we tend to just forget them over here, so we don't overwater them. We've got figs here, which are growing tiny little figs. I have made fig Newtons once from these plants. We've got here right now, in this little ball here, is basically a bunch of sphagnum. And what we did here, this is what we call air layering. It's a method for doing asexual propagation. And once you have roots there, you can just snip it off and plant it, and you've got yourself a new plant. Perfect genetic copy of the mother plant. Also in the room, because we're a small space and we kind of have to use the space efficiently to try to cram as much in as possible, we've got this weird looking device here that's not functioning right now. But in these buckets right now is cow manure. And we have a student that's taking this sort of, this product. It's an absorbent that's used in mining, but what they want to do is stick it in a pot with manure, and then see if the actual material will absorb anions. So if there was a soil amendment that you could use to hold on to your nitrates, your sulfates, and whatnot, all your sort of negatively charged nutrients, then they wouldn't be washed out of the soil so quickly. So this is a small little experiment going on here amongst our plant collection. This is kind of like a room where professors and students come if they don't have a space where they can, spread out and make a mess, essentially. Here we have a small student project. Holly, I can't remember Holly's last name off the top of my head right now, but she's doing a ethnobotany course where she's trying to make paper from some local plant material sources. So she's got some birch bark here, and she's soaking her samples for fibers right now here. Holly also reconstructed a calf moose skeleton in this space. So we have microscopes, we have scales for measuring, we have basically a fume hood over here, we have drying ovens, so say if you had some soil samples that you wanted to dry out. We've had students dry out wolf bone marrow in here. It stunk, it was terrible. But that's where people get sent when they have something stinky or something messy to do. They get sent up to the EFL and we make stuff happen. This space is just general use, and it can be used by undergraduate students, grad students, and professors. So yeah, you just have to come and contact us if you have need for space. And as you can see on the table here, we have stacks of what we call wood cookies. And this one here, you can see some time ago, something actually wounded the tree. And so you've got a scar here, and you can't really see the scar very well because of the duct tape here holding the sample together. But this particular project here, we're trying to figure out, when did the scars happen? and what caused the scars. And so using dendrochronology, we can actually put a calendar year to each particular ring using a method called cross-dating. This one here, you can, for all you foresters out there and people that know, and entomologists too, this is a pine tree. It was killed by mountain pine beetles, so you have that classic blue stain there. So we needed to first figure out when did the tree die, and then we can count back to that scar. You can get as fine as kind of dating the season of when the scar happened. So a lot of these scars are actually happening early spring. And so the samples were originally taken here to try to actually do some fire history, but we started to think that it wasn't necessarily fire. We're not sure, we're not totally certain, but I was talking to a few other people, like Roy Ray, for example, suggested that it could be actually rodent damage because they weren't, all the scars are really close together in years, but not the same year, exact same year. They're all early spring, when the tree would probably just be starting to grow and the cambium would be super lush, essentially, or, you know, the tree is starting to grow again. And the trees were not all that old. You can see that the tree was actually quite small when it was scarred. So we're starting to think that it's not necessarily a fire history, it might be a rodent history. So sometimes I've hired students to do work for me. I give them training in dendrochronology, and then I allow them to, like, we had a student measure in all these samples for us, for example. Say if you had an independent study, like in your 3rd or 4th year, where you wanted to do some tree ring research, this would be the place that you would come to do that. This is called the header house, and this is where the real dirty stuff happens. Like, we've got a very, very small wood shop where we can construct things for research purposes. We have a soil mixer for mixing up different soils. We have a soil pasteurizer here for, like, basically pasteurizing materials. You can set the temperature on this and leave it overnight. Over here we have a dust hood where you can do all your sanding. We like to do our sanding outdoors in the summertime, but in the wintertime sometimes it's too cold, so you can stand in here and sand your samples for the Dendrochronology Lab. If you have soil samples that you need to do shaking and sieving, we also have a soil shaker here that we use this structure for. The back here is kind of where we do a lot of our potting, transplanting, and the BC Timber Sales contract that I was talking about earlier. This is where we do all the assessments as well. You can see Jen here looks so happy. Doing some preliminary assessments on bud growth. So again, if you need a space where you need to do some dirty work, some sanding, soil shaking, build something in the wood shop, this is again where you would come. This is our office space, and we just wanted to let you know what John looks like with a mask on. This is the other greenhouse curator, John Orlowski. So John is incredibly knowledgeable in all things horticultural. Oh, that's a good question. John, when did the EFL get built? 2006?
No, in 2000. It opened in March of 2000.
2000. March 2000, if you didn't hear that. So, yeah, 21 years.
Yeah.
And exactly. John was the first greenhouse curator, and so I've been here 11 years, I guess. You know, like, we really like to see people, like, you know, find a supervisor, for example, find a research project, and maybe volunteer with the professor on a project that requires the EFL space, and then it'll just basically round out the experience so much more. I mean, we take care of the watering and all the propagation and stuff like that. That's kind of our job. And then the other part of our job is to assist people on those research projects. So if you can tie your volunteer work into an actual ongoing project with a professor, that is the ideal situation. And that's kind of like, then we'll be helping you as opposed to you helping us, you know, kind of thing. And that's kind of the way it should be. We're here for you guys. So, and then, you know, throughout the year, we'll hire students to work on, say, like the BC timber sales contract or maybe some dendrochronology work. So right now we have 3 students working in the lab. We had 4 up until last week. And so, yeah, that's basically all the staff that we'll have working here. We like to hire students that are like in their 3rd year, maybe their 2nd year, that are going to be around for a few years. And so it'll minimize the amount of training that we have to do. That work starts in January and goes till the end of March typically. So it's about 3 months work. It can be pretty go. So if you have a flexible schedule, or you you have maybe some larger blocks in your schedule, it can be sometimes like a good opportunity for you to like blend it in with your schoolwork. So there's that one, that 3 month opportunity from January to March. And then all the other stuff that we do that we hire students for is kind of random. So you just gotta like keep on coming in and seeing us and asking, you know, like, and maybe come for coffee and just and introduce yourself. That's always a good way to do it.